Thank you for joining the call today. I'm Catherine Maxey, Vice President of Public Affairs and Business Intelligence at Styron. And during the next hour, during our webinar, we're going to talk about the safety of polycarbonate, specifically in medical applications. As you probably know, polycarbonate is a very valuable plastic with a unique set of properties that are highly valued for a variety of applications, including medical devices. And the topic of the safety of polycarbonate has been a source of some controversy because of the presence of bisphenol A, which of course is a monomer used in the polymerization of polycarbonate. BPA, of course, is present in the polymer at very low residual levels, and this has raised some questions about the level of BPA that might migrate from the plastic and whether or not there are health and safety issues from any potential leaching that may occur. Many claims are out there, but the weight of evidence and the largest, most comprehensive studies show no health effects. So today we'll be talking more about that, and we're bringing you two experts on different aspects of the science around the safety of BPA. So now let me tee up our topic for today, and we'll go to the next slide. To set some context as background, polycarbonate has been used in a variety of applications for consumer and industrial products for decades. In medical device applications, polycarbonate is also valued highly. Its properties include strength, toughness, rigidity, clarity, resistance to sterilization, and heat tolerance, all properties that are needed in many types of medical devices today, and properties that can't be compromised. And during this time, many studies have been conducted to ensure that polycarbonate is safe. In fact, BPA is one of the most thoroughly tested chemicals used today. Government and scientific bodies around the globe have extensively evaluated the weight of evidence of BPA and have declared that it's safe as used, including in materials that come in contact with food. And because of the weight of scientific evidence demonstrating this point, there's been a strong consensus authorizing polycarbonate for a variety of uses. In fact, none of the many hundreds of studies on BPA have shown a direct cause and effect relationship between BPA and any human health effect. And in September 2011, as an example, an international panel of experts organized by the World Health Organization and the Food and Agricultural Organization of the United Nations released a report on their review of all the latest scientific evidence on BPA and concluded that initiation of public health measures would be premature. But nevertheless, BPA remains a hot topic, and that's probably why you're here. So with that as an introduction, let me introduce our presenters who are going to be sharing some great research with us today. We will be hearing from Jackie Anim from Ethicon Surgical Care. And Jackie is the staff subject matter expert of polymer plastics and materials at Ethicon, which of course is a manufacturer of medical devices for a host of healthcare organizations. Since Ethicon is a major user of polycarbonate, they wanted to increase their own understanding of the safety of the material and conducted some research around the migration of BPA from polycarbonate, and she'll be telling us more about that. We'll also hear from Dr. Steve Henches, who's the Executive Director of the Polycarbonate BPA Global Group at the American Chemistry Council. And Steve will share the toxicological and epidemiological research on polycarbonate and BPA with a particular focus on the safety of BPA. And he'll also highlight some studies that are soon to be published. And as I said, I'm Catherine Maxey, Vice President of Public Affairs and Business Intelligence at Styron, and we're the sponsor of today's webinar. Polycarbonate is part of Styron's plastics portfolio, and we closely monitor BPA research and findings. And our Styron scientists, like our two presenters, work with polycarbonate. We feel very confident about the science and the safety of polycarbonate, but we recognize the challenges that you face in sifting through all the information about BPA that's out there. So we hope this webinar is helpful and clarifies some points for you. Now, before I turn it over to our first speaker, we want to first ask our first polling question. And we'd like each of you to answer the question that we're showing here. Which best describes your use of polycarbonate? So please go ahead and click your answer, and we'll give you just a minute, and then we'll show the group's answers in a graph. All right, it looks like most of you have voted. We've got almost 100 people on our webinar, and more than 3/4 have voted. So let's go ahead and put up our graph. That's great. So roughly 2/3 of you are using polycarbonate and plan to continue to use it for its unique properties. That's great to hear. 17% are thinking about switching, so hopefully today's webinar will help you with that decision and understanding more about polycarbonate. And we also have some folks that are not using it today and some from outside the industry. So that's very helpful. Good to understand kind of where everyone's at. I want to turn it over to Steve Henches from the Polycarbonate BPA Global Group. Steve's going to share an update on the recent research that's been done and give us a look at what's on the horizon. So Steve, over to you.
Thank you, Catherine, and welcome everybody to the webinar. Thanks for joining. In the next 15 minutes or so, I'll provide a brief overview of BPA, including how it's used, the views of many government agencies on its safety, the science underlying those views, and a few notable things coming up, things that we can look forward to. So if we can move to the next slide, we'll start with what is really some old news, just to be sure that we're all on the same page. As you may know, most BPA is used to make two things. It's used to make polycarbonate plastic, and it's also used to make epoxy resins. Most of you on the call, on the webinar, probably recognize polycarbonate. It's a clear, very highly shatter-resistant material that's very widely used in common consumer products. Things like reusable food and beverage containers, sports safety equipment like bicycle helmets, CDs and DVDs, and many other products that we use in our daily lives. It's also used quite widely to make critical components of many life-saving medical devices, which are the focus of today's webinar. Epoxy resins are quite different materials. They are very widely used as protective coatings, and you'll find them quite commonly in many food and beverage cans where they protect the safety and integrity of the contents. They're also used as structural steel coatings, things that you can't really see, but they're very important. And they're also used widely, for example, as the primer coat on automobile bodies. You'll also find them in other applications like adhesives and laminates. A very important attribute of these materials is that only trace levels of residual BPA remain in finished products made from polycarbonate or epoxy. Typically, these materials contain less than 50 parts per million of residual BPA. And what that means is that there is only very limited potential for exposure to BPA from use of products made from polycarbonate or epoxy. Over its long history of 50 years of use, BPA has become one of the best tested substances in commerce. There are literally many, many hundreds of studies on BPA that have been published in the scientific literature. And what that means for us is that we have a very rich scientific database that's available to assess the safety of BPA. On the next slide, we'll take a look at what government experts have concluded after reviewing that scientific database. In recent years, quite a few government agencies and expert scientific panels have very extensively reviewed the research, the science on the safety of BPA. At this point, the consensus of many government agencies around the world is that BPA is safe as it is currently used in its many applications. And what I show on the middle of this slide is a quote from FDA's website. This is an updated perspective from FDA that was just posted at the end of March of this year. FDA in the U.S. is very important because FDA regulates both food contact products as well as medical products. And as you can see here, FDA states that BPA is safe at the very low levels that occur in some foods. They also go on to say that that perspective, that view, is based on their own review of hundreds of studies on BPA, including the latest findings from new studies initiated by the agency. Although it's more than we can really go into today, I think it's important for you to know that over the last several years, FDA has embarked on a very comprehensive set of studies on BPA, studies being conducted in their own laboratory. To date, 13 of those studies have been published in the peer-reviewed scientific literature, and those studies collectively provide very strong support for the safety of BPA. Similar conclusions have been reached by other government agencies around the world, notably the European Food Safety Authority. Up in Canada, Health Canada has reached a similar conclusion, and also over in Japan, the Japanese government agencies. Most of these reviews and conclusions are focused on food contact applications because that's where most of the controversy about BPA has been. But the underlying science that supports these conclusions is applicable to medical devices as well. On the next few slides, we'll take a quick look at some of that key science, starting on the next slide with the subject of pharmacokinetics. Pharmacokinetics describes the processes by which a substance is absorbed into the body, where it goes once it's in the body, what form it's in, is it metabolized, how is it eliminated, and how long does it stay in the body. Although pharmacokinetic studies do not directly measure toxicity, they do set limits on the potential for a substance to be toxic. Since most toxicity studies on BPA have been conducted by the oral route of exposure, which is most directly relevant for food contact products, pharmacokinetic data is also critical to assess the safety of other products other than food contact, in particular medical devices. Several clinical studies on human volunteers have confirmed that BPA is very efficiently converted to a biologically inactive metabolite after oral exposure. This is a very important point because, as you may know, BPA itself is weakly estrogenic, but it's been shown that the metabolite is biologically inactive, and in particular, it's not estrogenic. In a study published in 2011, this feature was illustrated quite well. A group of human volunteers were treated with a diet that was atypically high in BPA content, yet no free BPA could be detected in blood, even with a very sensitive analytical method. Only the metabolite could be found in blood, and even that was present only transiently. The reason is that the metabolite is rapidly eliminated from the body. The half-life is only a few hours, and it's eliminated in urine. What that means is that if we are exposed to BPA, it's eliminated from the body within the day of exposure. It doesn't remain in the body. It doesn't accumulate in the body. These points have been illustrated and extended in quite a few studies on laboratory animals, in particular non-human primates or monkeys, as well as rodent studies. These studies have shown that the efficient metabolism of BPA that has been shown to occur in human adults also occurs at all ages, not only in adults, but also in pregnant animals, in neonates or infants, and also in the fetus. And what this data suggests is that there should be no special sensitivity to BPA at different life stages. BPA has also been shown to be efficiently metabolized and eliminated from the body after IV, intravenous exposure. And those studies also show that even with IV exposure, BPA doesn't accumulate in the body. It's very quickly eliminated. That data is very important, in particular for medical device applications that involve exposure to blood. So overall, these studies show that BPA is very efficiently metabolized. It's eliminated quickly from the body. And we would predict, based on this data, that BPA should have low toxicity. On the next slide, let's take a quick look at what the experimental toxicity data actually tells us. As you may know, much of the controversy around BPA is due to its weak estrogenic character. And so the controversy has been that BPA could cause endocrine-mediated effects at low doses. There are many claims about BPA, claims that BPA can cause reproductive or developmental effects, neurobehavioral effects, and even cancer, as well as other claims. But if we look at the most comprehensive studies on BPA, a different story emerges. For example, multi-generation reproductive and development studies have consistently found no low-dose effects. With most chemicals, we'd be lucky if we had one multi-generation study, but in the case of BPA, multiple studies, multi-generation studies have been conducted, at least 5 very high-quality studies. A couple of those have been sponsored by industry, but the others have been sponsored by government agencies. And these studies consistently show the same thing. No effects have been found at low doses. And that is true for multiple strains of rats and mice, which gives us confidence in the validity of these results. Similarly, FDA has recently conducted a very large-scale, very comprehensive subchronic study, and likewise, they found no low-dose effects. They found effects only at very high doses, as would be expected. Neurobehavioral effects have not been found in separate studies conducted both by FDA and EPA in their own laboratories. And regarding cancer, no clear evidence of carcinogenicity has been found in lifetime studies on both rats and mice. These are studies conducted by the U.S. National Toxicology Program. So based on all of that data, we can confirm that BPA, as expected, does have low toxicity. And based on all of that data, government agencies have established safe intake limits of 25 to 50 micrograms of BPA per kilogram of body weight. To understand safety or risk, though, we also need to know how much BPA people are actually exposed to. And we'll take a look at that on the next slide. We know that BPA has a very short half-life in the body. This is coming from the pharmacokinetic studies. We also know that it's entirely excreted in urine in the form of a metabolite. Accordingly, urine biomonitoring provides the best way to directly measure human exposure to BPA. In the U.S. and Canada, population-scale urine biomonitoring studies have been conducted. In the U.S., they're conducted by the CDC. These studies show that median or typical intake exposure to BPA is less than 50 nanograms of BPA per kilogram of body weight. That's the usual metric for exposure. If you think back to the safe intake limits, 25 to 50 micrograms of BPA per kilogram, we see that typical human exposure is about 300 to 1,000-fold below the safe intake limits, not even remotely close to those safe intake limits. And you should also know that the safe intake limits already have a 1,000-fold safety factor built in. Low exposure has been corroborated by the low levels of BPA that is present in polycarbonate products and also epoxy resins, as mentioned earlier. Quite a few studies, migration studies, have been conducted both on polycarbonate and also on epoxy. These studies show that very low levels of BPA can migrate out of these materials. And the levels that migrate are consistent with the levels that are then found in the urine biomonitoring studies. We would also expect low migration from polycarbonate medical devices, but few studies have been published in the scientific literature to confirm that point. And that's why the study that you're going to hear about from Ethicon in a few minutes is very important. So let's go to the next slide, and we'll take a look at a few things that are coming up, a few important events that are coming up. We do expect more reviews from regulatory agencies around the world. We expect more updates from FDA. Recall that their last update was at the end of March this year, but we do expect further updates from FDA as additional research on BPA is published, in particular as their additional research is published. We also expect a draft report from the European Food Safety Authority in July of this year, with a final report to follow in November of this year. And we are anticipating a draft report on the safety of BPA in medical devices in November of this year. That report is coming from a scientific committee in Europe. You see the acronym on the slide. It is the Scientific Committee on Emerging and Newly Identified Health Risks. So that draft report should come out in November of this year. We also expect more important government lab research, and we expect that that research will further reduce any uncertainties about the safety of BPA. I mentioned an FDA subchronic toxicity study. We're aware of that study from presentations at scientific conferences, but the study hasn't been published yet. We expect that it will be published very soon, and we're very much looking forward to the results of that study. FDA is now conducting a chronic toxicity study that has been initiated, but that's a multi-year study, so we're not going to see results from that for several years now. Although several human pharmacokinetic studies have already been conducted, the U.S. National Toxicology Program is currently conducting what will be the most comprehensive study to date. And we anticipate, based on what we've heard so far, that that study will confirm what we know from already available pharmacokinetic studies. And finally, we're aware that FDA is also conducting 2 exposure studies on medical devices. We haven't seen results from those studies yet, but we're looking forward to those studies sometime in the future. So in conclusion, based on everything we know about BPA, based on all the science that we have available to us, we are confident that polycarbonate and epoxy resins are safe as used in food contact applications, and that polycarbonate is safe as used in medical device applications.
Okay, great. Thanks, Steve. That was a wonderful overview of a very large and complex issue, so thank you for doing that. During the Q&A session, you'll have an opportunity to ask questions of Steve and Jackie, and you can actually type in questions at any time or at the end. We'll be addressing all the Q&As at the end. And this presentation will be shared with all of you through an email that will be sent out after the webinar. So you'll be getting a link to a recording of this program. So let's go on to our next slide. Before we bring on Jackie, we're going to have another one of our polling questions. And for those of you who weren't on when we started, this is just our chance to hear from you and to have you answer some interesting questions we've got here. Our next polling question is, please indicate your level of concern about polycarbonate. So go ahead and click your answer, and we'll just take a few moments for everyone to participate in the poll, and then we'll show you the results. So the survey is showing that, well, the vast majority of you feel that polycarbonate is safe. 38% saying it's safe and you don't have any concerns. 43% saying it's generally safe, but you have some concerns. So it's great that we're having this webinar today to hopefully address some of the concerns that you have. 13% undecided, and 4% have concerns. So, or 5% rather. So thank you for that poll. And now let's bring up our next presenter, Jackie Aneem from Ethicon. So Jackie, I'll turn it over to you now.
Thank you, Catherine. Again, my name is Jackie Aneem, and I will be presenting the outcome of the analytical study conducted by Ethicon Surgical Care via a third-party laboratory house, NAMSA. The title of my presentation is Residual Bisphenol A Content in 3 Sources of Polycarbonate, the Analytical Story. On the next slide, I will review the table of contents, starting with a brief overview, followed by background and scope of the research, defining our focal point, and spend majority of my time focusing on results of the analytical investigation, and finish up with a short paragraph of acknowledgement to the Ethicon Surgical Care BPA investigation group located in Cincinnati, Ohio, with a special thanks to our sponsor, Sam Onukuri of MDMD. Next slide. Global purchasing organizations such as Premier Health and Health Trust are now making purchasing decisions based on the potential presence of listed chemicals in product. And most of the time, binary decisions are required of us with no middle ground or use of explanatory facts. Tools such as decision trees have been developed internally to help maintain the BPA level to a level below the current EPA tolerance, which is 50 micrograms per kilogram per day. In order to fully understand the scope of the concern, we embarked on this analytical study for answers. Next. Now, so far as BPA is concerned, there's always been some discussion around the world relative to its safety. But recently, there's been a strong agenda to design BPA out of commercial product due to concern over potential biological impact. Research to date has focused on hormone mimicking effect, but the results remain controversial. One thing that the industry sees as a gap is quantitative analysis of the actual amount in specific material and its sensitivity to environmental factors such as molding and sterilization. So the intent of this presentation is to help bridge the gap. Next. The scope of the research is the utilization of analytical technique to quantify the exact amount of bisphenol A in 3 sources of polycarbonate. The test protocol included the investigation of secondary operations such as injection molding and gamma sterilization techniques. Gamma was selected due to its widely used in medical industry as most preferred sterilization method due to its cycle time and cost benefits. Results were then compared to the US EPA reference dose, which is the same as the EU tolerance daily intake of 50 micrograms per kilogram per day. Next slide. So far as the analytical study is concerned, factors such as type of polycarbonate, which came in the form of medical, automotive, and consumer grade, and the consumer grade was actually a drinking cups that were purchased from off the shelf. Impact of manufacturing processes such as injection molded versus pellet, gamma sterilization versus no sterilization were reviewed. 3 extraction techniques were utilized in this research, and they included isopropyl alcohol, ethanol alcohol, and exhaustive extraction by Soxhlet. My next couple of slides will showcase the results from this study. The IPA extraction results indicated, with the exception of the drinking cups, which is the consumer-grade polycarbonate, BPA concentrations were well below the level of detection. This was evident from the rest of the data. In fact, the amount was less than 0.5 microgram per gram sample. BPA found in the drinking cups was 5.8 microgram BPA per gram for the unsterilized condition versus 5.3 microgram BPA per gram sample for the sterilized equivalent. Next slide. So, assuming the exposure of 10 gram of polycarbonate, which could come in the form of a medical device, and a weighted adult of 70 kilogram, the worst case of human exposure will be the 5.8 times the weight of the device divided by the body mass of the individual. And this will translate to 0.76 microgram per kilogram for the sterilized drinking cup and 0.83 microgram per kilogram for the unsterilized equivalent. However, the US EPA reference dose today is 50 microgram per kilogram per day. Data also showed that the sterilization method did not impact the results significantly. Additionally, when it came to the ethanol extraction via HPLC, the trend was very similar to the results described previously, in that, with the exception of the sterilized drinking cup, the BPA concentrations were well below level of detection. The reported values were less than 0.5 microgram per gram polycarbonate. However, the exact concentration of the BPA found in the sterilized drinking cup was 4.0 microgram BPA per gram sample. This value could be converted or normalized to the same unit described by the EPA on the next slide. Using the same argument and algorithm, and assuming a 10-gram exposure to a medium-built adult of 70 kilogram, the worst case of human exposure with the 4.0 microgram BPA per gram translate to 0.57 microgram per kilogram per day. Next slide. Unlike the IPA extraction and the ethanol, the exhaustive extraction by Soxhlet was conducted just on the medical grade polycarbonate. The intent was to improve the level of detection by utilizing the most aggressive technique via analytical method and combined with IPA. Medical grade was selected due to its linkage to our business model. And with that, the average BPA found to contain in the resin prior to injection molding was 0.25 microgram per gram polycarbonate and 0.28 microgram per gram after the injection molding. Next slide. These numbers translate to 0.036 microgram per kilogram per day and 0.04 microgram per kilogram for the injection molded option using the same argument and mathematical model as previous cases. Next slide. In conclusion, the IPA extraction condition was designed to simulate condition encountered during normal human contact. The ethanol extraction was quite aggressive, using increased temperatures of 50 Celsius versus 37 for the IPA extraction, as well as concentrating the volume of the extract. But the exhaustive extraction by Soxhlet was the most aggressive condition within the study. The results from the leaching could be described as the most extreme worst case of human exposure. Next slide. As a note, sterilization did not seem to impact the medical and automotive grade polycarbonate, but the consumer grade did, however, show significant shift when the drinking cups went through sterilization followed by ethanol extraction. This behavior could be explained from the fact that most consumer grade material may not have been formulated for gamma resistance. Next slide. Also, IPA extraction could be an alternative method for determining the BPA content of polycarbonate if the detection level could be enhanced. the amount of BPA in 10-gram sample of the medical-grade polycarbonate will result in a worst-case human exposure of 0.04 microgram per kilogram per day. When this value is compared to the current reference dose from the EPA, which is 50 microgram per kilogram per day, the worst-case exposure from this grade of material is 1,250 times less than the prescribed value from the EPA. Next. From a molder perspective, BPA found in medical-grade polycarbonate after molding was 0.28 microgram per gram BPA. The worst-case exposure for this level of concentration will be 0.04 microgram per kilogram per day, and this is 1,250 times less than the US EPA reference dose. Additionally, the automotive grade was 660 times less than the same reference dose, and the consumer grade was 16 times less than the same standard as prescribed by the US EPA. And in final, I would like to finish my presentation by stating that all three polycarbonate did in fact contain some residual bisphenol A. However, the level was well below the EPA reference dose for safety. At this point, I would like to express my gratitude to the EPA investigation team at Ethicon Surgical Care, including Jeffrey Daniel, Chris Jones, Pamela Attenweiler, Bob Prisgoda, as well as Sam Onukuri of MD&D for funding the project and the test staff at NAMSA. Thank you very much, and at this point, I'd like to hand it over to Catherine.
Great, thank you so much, Jackie. And again, we'll have time for Q&A at the very end, so if you have questions, you can go ahead and type them into the questions section on your webinar screen at any time. But before we get into the Q&A, I want to try to summarize in sort of a conclusion a few slides here, and then we'll have our final polling question in our Q&A. So next slide, please. So from our perspective, the challenge now is to overcome the misinformation that's out there about BPA in the marketplace. As you heard today, major government agencies worldwide have declared BPA as safe as used in many applications. And these conclusions are based on numerous scientific studies and supported by a variety of scientific organizations. And more research, some very comprehensive studies, including some from the US government, will be published soon and are expected to confirm this position. We also know that when it comes to exposure, the amount of actual BPA that people are exposed to is far below the levels of safe exposure set by regulatory bodies. So when you think about the balance of information we've described today, we also know that there are contradictions out there to these scientific research findings that are being published all the time that have suggested that to the public that polycarbonate and BPA may be unsafe. Because of this, decisions may be made based on erroneous information, and polycarbonate often could wind up being deselected. Now, these replacement materials typically have lesser attributes than the proven performance of polycarbonate. And candidly, we don't want to introduce materials for patient care that may not be quite as safe and as well tested as polycarbonate is. Next slide. So for medical device producers, there's many considerations. We all know that in the healthcare field, bringing life-saving and life-enhancing products to market requires more than just medical breakthroughs. It requires materials that can meet the rigorous challenges that these new applications demand. And polycarbonate is a proven material with significant benefits. You can see they're listed here on the screen. And they really enable some of the advanced procedures that are common today, whether it's non-invasive surgeries, dialysis treatment, endoscopic procedures, etc., with polycarbonate serving as a critical component in many devices, allowing medical professionals to do things like directly observing blood and other fluids in life-saving procedures. So we don't want to see these attributes compromised or make a concession to other materials that are not as proven as polycarbonate is. So with that, let's take our last polling question before we move into our Q&A segment. You can see the question up on your screen now. Please take a moment to click on your answer as to whether you plan to use polycarbonate in the future. And now you can see our survey results with a very large group of people planning to continue to use polycarbonate. That's great. Some people still thinking about switching, some not planning and not currently using and not planning to use, and a group of people who are not in the resin selection role. So thank you very much for participating in our poll. And let's switch over to our Q&A. It looks like we've already received some questions here. And let me take a quick look through these. And Steve and Jackie are on the line ready to pick up and answer some of these questions. So my first question I think is aimed for Steve, and the question is, can you comment about the regulatory situation in France? Specifically, will this affect medical devices in France or Europe? Steve?
Okay. In France, at the moment, I don't think the regulatory situation will have any impact on medical devices. There was a proposal about a year or so ago that would have affected not only BPA, but actually a wide range of materials in medical devices. That proposal, however, it was a legislative proposal, not regulatory. That proposal, however, did not pass. So what is currently in play only affects food contact products, and we're not at the end of that process to even know if that will ever happen or not. But at the moment, there is no impact on medical devices that I know of.
Okay, great. Thanks. Also, a related question here, Steve, about can you comment about the current Proposition 65 issue in California?
Well, that's an ongoing process. Actually, it's been ongoing for about almost 5 years now. And I won't give a long summary, but just a couple of highlights. Way back in 2009, the expert committee in California voted unanimously that BPA should not be listed under Proposition 65. More recently, the state agency, OEHA, determined that it could be listed under Proposition 65, so we have differing views. And to resolve that, we filed a lawsuit in California, and that lawsuit is just in the very early stages. So at the moment, BPA is not on the Proposition 65 list, and there will be no final action until the lawsuit is resolved, which I can't predict how long that could take, but it'll take some time for sure.
Okay, great. A couple questions here about occupational exposure. One person asking about can BPA become airborne when it's processed in pellet form with exposure through breathing. Other question being about can BPA be absorbed in the skin when an employee is handling warm parts after being molded. So I don't know if maybe we can throw this out to both of you to talk about occupational exposure.
Yeah, I'll jump in and start. Those are very good questions. They're ones that probably haven't gotten a lot of attention that I've seen, but there is some relevant data. Regarding inhalation when polycarbonate is processed, I'm aware of 2 relevant studies. One wasn't really looking at worker exposure, but it was looking at emissions of anything, including BPA, from the extruder when polycarbonate is being processed. And recall back to my presentation, polycarbonate only contains trace levels, less than 50 parts per million typically, of residual BPA, so there isn't much there in the first place. And the study looking at extruder emissions confirmed that even when being processed at the high temperatures needed to process polycarbonate, very little BPA was coming out of the plastic in the extruder emissions. So that didn't look directly at worker exposure, but would suggest that there isn't going to be a lot. The second study is much more on target. It was actually a biomonitoring study that looked at worker exposure to BPA, workers who were in the facility processing polycarbonate. What that study found is that worker exposure was not all that different from the background exposure to BPA, just from use of consumer products, for example. And again, if you think back to my presentation, background exposure to BPA is extremely low. So this other study indicated that worker exposure when processing polycarbonate would also be in the range of background, not so different. Regarding dermal exposure, I'm not aware of any studies that have looked at dermal exposure from handling polycarbonate, materials. But again, keep in mind that the level of residual BPA is extremely low, less than 50 parts per million. And it turns out that BPA is not easily released from the plastic. The diffusion of that residual BPA out of the plastic does not happen very easily. So the potential for dermal exposure is going to be very low. And then in addition, other studies have shown that when BPA is applied to the skin, it does not readily go through the skin. So although we don't have any studies that I'm aware of that looked at dermal exposure from handling polycarbonate, the evidence that we do have would suggest that worker exposure would not be significant at all from handling polycarbonate articles.
Jackie, anything to add?
Yeah, what I was going to add is the bisphenol A is very active and migratory when the polycarbonate is heated. So far as skin contact and dermal exposure, just like Steve had indicated, it will be very, very minimum. There's limited data to speak to at this point, but that's an interesting question that needs to be tracked as we move forward.
Okay. Next question I have is the BPA metabolite complex active in the body?
Okay, that's probably for me.
Yeah.
Is the metabolite active in the body? The answer to that is the metabolite formed from BPA has no known biological activity, and it's specifically been tested to determine whether it's estrogenic. The reason being that BPA itself is weakly estrogenic. It has very low potency, but it is weakly estrogenic. The metabolite, on the other hand, has been found to be not estrogenic at all. So the metabolite has no known biological activity. And in a roundabout way, that really has been confirmed. If you think about the most comprehensive toxicity studies, since BPA is actually metabolized as it enters the body, in a sense, those studies really are toxicity studies on the metabolite. And so from the most comprehensive studies, we find out that BPA and therefore its metabolite have very low toxicity. So no known biological or significant biological activity with the metabolite.
Okay, great. There's a question here about, can you comment regarding the 0.1% requirement from REACH and how to assess this data? It says 0.1% W/W. I'm not sure what that is abbreviation for. Does that sound familiar at all, Steve?
Vaguely, and I think I'm probably not able to give a coherent answer, but I think that's when I would suggest maybe we can follow up and give a better answer offline.
Okay. All right. Sounds good. Got a question here about the studies that you mentioned that are pending and that will be published in the coming months or years. If you can provide a little bit more information there about the FDA subchronic toxicity study that was reported at a scientific conference, and also the NTP human study, if it's going to address blood exposure.
Starting with FDA's subchronic study, that has been completed and has been reported at actually 3 scientific conferences. So we have a pretty good idea about the findings of that study, and we hope it'll be published soon. It was a very comprehensive study. It involved 9 dose levels of BPA, ranging from very low, the ones that are most relevant for human exposure, all the way up to very high dose levels. The study also involved 2 doses of what is called a reference estrogen to make sure that the researchers could know what to look for with an estrogenic compound in that study. And what they reported at the scientific conferences is that they found no effects at any of the low doses, any of the 7 low doses, including the lowest ones most relevant to human exposure. They found only effects at the 2 very high doses, that would be expected based on other studies. Any compound would show effects if the dose is high enough, and that's what they found in this study. So although we don't have complete details, only what has been presented at the scientific conference, we're very interested to see this study published. And I think we can speculate, at least, that the study had a lot of influence on FDA's most recent perspective on BPA, where they stated very clearly that they believe BPA is safe at the very low levels that are present in food. Regarding the NTP human study, again, this is underway, so we don't know a lot about it. We know that the study is starting off with oral exposure, which is, of course, quite relevant because that's where most of the controversy has been. But we also know that NTP is considering extending the study to also look at dermal exposure and IV exposure. The latter is the one that would be most relevant to medical devices that contact blood. I don't know, I have not heard yet whether they will actually do the dermal and IV studies, but I believe they will. My understanding is they will proceed to those. Regardless of whether they do or don't, there's plenty of laboratory animal data that involve IV exposure. So we have studies both in monkeys and in rodents. That data collectively will be invaluable to FDA and others as they evaluate safety of BPA in medical devices. So the NTP study, if they do it, would be kind of like icing on the cake, but we already have a lot of very good data.
We got a question about safety of polycarbonate in relation to neonates and small children.
That one's probably for me also?
Yeah, I think so.
Yeah, and that's where most of the controversy about BPA has been at developmental stages of life, neonates, infants, and during pregnancy for that matter. Those are the stages where you would be most interested in looking for health effects, and that's where most of the research has been. So starting with the pharmacokinetic data, again, what we're looking for there is whether BPA is efficiently metabolized and cleared from the body. Are there any life stages where that doesn't happen or doesn't happen very well? What FDA's research, as well as other studies, have shown is that even just after birth, BPA is efficiently metabolized about the same as it is in adults. This has been shown in one of FDA's studies on monkeys, and similar results on pregnant animals as well. Those, of course, are studies that you can't conduct in humans, so the monkey data is very important. So we don't have any indication that at developmental stages that BPA should be more sensitive, or developmental stages of life would be more sensitive to BPA. Most of the toxicity data also then focuses on developmental stages from pregnancy on through young ages and development. And going back to the more comprehensive studies, in particular the multi-generation studies, we find in those studies consistently no effects at low doses, and that does include the sensitive stages of pregnancy and early development. So if we look at all the data together, I think we're confident that BPA is safe even at those developmental stages, and certainly agencies like FDA have carefully considered that issue in reaching their conclusion.
I think we just have time for one last question, and then we'll have our final poll. The question here is, can you recommend some additional in-depth resources that I can share with my colleagues, perhaps some recent publications or websites?
A couple of things I can offer, Catherine. I think you may have already mentioned this, but some additional materials will be available along with the presentation. I'm not quite sure how those are distributed on the webinar, if they're sent out or available through the webinar site. But a number of fact sheets and other materials will be made available to participants. Also, one of our websites, www.factsaboutbpa.org, that was shown at the end of my presentation, www.factsaboutbpa.org, is another resource that we have available with info on BPA. We're currently updating that site, so if you look at it now, you'll see a pretty good site, and if you come back in a month or so, you'll see an even better site. So visit now and visit often.
Okay, great. Jackie, anything to add as far as any resources?
What we've done so far has always been kept internally. However, if anyone needs additional information relative to the test method and details on how those samples were prepped and tested, we're willing to share that level of detail offline.
Great. Thank you so much, Jackie. I'm sure you'll get some follow-up questions about that. So great Q&A session. Lots of really good questions that came in. If we didn't have time for your question, I apologize for that, but we will, as we said, be sending some follow-up resources and can exchange information with you that way. And at this point, we just want to say thank you for joining our webinar today. We had a great turnout. It's obviously a topic of a lot of interest, and hopefully we were able to answer some of your questions and concerns. So thank you for participating in our webinar today, and have a great day.